Bismillahirrahmanirrahim. This video is about the molecular basis of cancer, and one of the hallmark is unregulated cellular proliferation. These are the references for this video.
The cell proliferates as it receives signal, and that signal is processed through various mechanisms. But tumor cell is self-sufficient in growth signals. Proto-oncogene is a gene, a normal gene that provides signals that lead to cell division or apoptosis, while the oncogenes are mutated genes that cause excessive cell growth even in the absence of growth factors and other growth promoting external stimulus. The oncoprotein is a protein that is coded by an oncogene, and this oncogene, which has been integrated into the genome of a eukaryote, and that is involved in the regulation or synthesis of the protein linked to tumorigenic cell growth. The mutation through various mechanisms lead to altered functions of the oncoprotein, and these are constitutively active, and these are ligand independent and resistant to control by external stimulus. And so this mechanism will lead to the independence of the cell from normal checkpoints that will result in excessive proliferation. The cell is under strict control by various mechanisms we have discussed earlier, and by numerous mutations the cell gets itself free from these control and rapidly proliferates. These mutations can occur in activating gene or inactivating genes. So the oncogenes are the genes that stimulate passage through the cell cycles, while inactivating genes, they prevent the inhibitory influences of the tumor suppressor gene. Hence, they also increase the cellular proliferation because they inhibit the tumor suppression gene. There are various mechanisms that stimulate the cell to proliferate. The component of these mechanisms are growth factors, cell surface receptors, intracellular signal transduction pathways, and DNA binding nuclear proteins or transcription factors. How this growth factor induced signaling exerts its effect. It binds to its specific receptor. There is a limited activation of the growth factor receptor that activate several cytoplasmic signaling transducing proteins. The transduced signal is then transmitted to the nucleus via effector protein, second messenger, or a series of signal transducing molecule. Then there is induction and activation of transcription factors and simultaneous epigenetic alteration that initiate and keep up the DNA transcription. The genes are expressed and encode factors that promote entry of the cell into cell cycle that will result in cell division. Simultaneously, other genes that are responsible for cell survival and metabolic alteration are also expressed. This illustration is summarizing the signaling model in cellular transformation. A ligand binds with the cell membrane receptor. The examples of ligand are cis, HST, androgen, and estrogen, and the receptors are EGFR, ERB2, and KIT. When this ligand binds to its receptor,
specific receptor, the receptor can activate intracellular signaling directly, or a protein binds to the activated receptor, and this activated protein binding couple may then trigger intracellular signaling. The example of these proteins are SRC, LCK, NES. Or the receptor is G protein coupled that stimulate guanine nucleotide related signaling, and the important related molecules are RAS, MOS, and heterotrimeric G protein. Or the ligand may traverse the cell membrane to activate the receptors that are present within the cytosol. There is no cell membrane intermediate, and there is a direct activation of the receptor that is present within the cytosol. The examples of these receptors are PPAR, androgen and estrogen receptors. When there is activated receptor and activation of the receptor, there is then intermediate signaling activation, such as BRAF and MAP kinases activation, and after that the activation of transcription. Important transcription factors are MIC, FOS, JUN and MIB. This picture is from chapter 1 of the Robbins basis of disease, and it is showing various signaling pathways which are responsible for receptor mediated signaling. The various type of the receptors are present on the cell or in the cell, and they include a non-receptor tyrosine kinase based receptor, a tyrosine kinase receptor, a nuclear receptor that binds with its ligand and then can change the transcription, a 7-pass transmembrane receptor linked to a G protein notch which recognizes a ligand on a cell and is cleaved, and then it yields an intracellular fragment. This intracellular fragment then can enter into the nucleus and change the transcription of specific target genes. Then vent or frizzled pathways where activation releases intracellular beta-catenin. This intracellular beta-catenin is from a protein complex, multi-subunit protein complex, and the release of beta-catenin then can migrate to the nucleus, and this beta-catenin act as a transcription factor. The low density lipoprotein LDL receptor related protein 5 and 6 are homologous, and they work as a co-receptor in vent frizzled signaling pathways. Proliferation is under the control of the two forces. One are the forces that drive the cell to divide, while the other forces control the cell cycle, and these regulators are the growth factors, cell surface receptor, intracellular signal transduction pathway, and DNA binding nuclear protein that are transcription factors. When cell get rid of these regulators, or there are tumor driving mutations, there is the proliferation of the cell or division of the cell that is not dependent on the regulators such as the growth factor and the signal transduction, etc. Actually, there are proteins that are produced, and these drive now the cellular proliferation without the normal restraint. Ligand is something that binds to a biologic molecule, and this ligand attaches to its specific receptor and forms a complex, as we have seen earlier. A ligand is a continuous need of normal cell and may be a need of a developing tumor. Some ligand they drive cell multiplication in early oncogenesis. Later, this cell become independent of ligand, and this is made possible by changes in receptor and other molecules. While some of the tumor they produce ligand themselves, and there is autocrine function of these ligand on the tumor cells, and they multiply. The example is the oncoproteins that are produced as a result of gene amplification. The aberration or deviation are seen in growth factor receptors, and other factors which are responsible for cell cycle regulation, and these proto-oncogene are associated with a specific type of tumor, such as platelet derived growth factor beta is overexpressed in astrocytoma, while fibroblast growth factor, one of this is HST1, that is also termed as fibroblast growth factor 4, and it is overexpressed in osteosarcoma, while FGF3 is amplified in stomach cancer, bladder cancer, breast cancer, and melanoma. The transforming growth factor alpha is overexpressed in astrocytoma, while hepatocyte growth factor is overexpressed in hepatocellular carcinoma and thyroid cancer. Similarly, the growth factor receptors are also deviated, and the epidermal growth factor family member ERB1 is mutated in adenocarcinoma of lung, while ERB2 amplification is seen in breast carcinoma. FMS like tyrosine kinase 3 receptor, that is receptor for macrophage colony stimulating factor, it shows point mutation or small duplication and is associated with leukemias. Receptor for neurotropic factors, the proto-oncogene is REAT, and it shows point mutation in multiple endocrine neoplasia 2A and 2B and familial medullary thyroid carcinoma. Platelet derived growth factor receptor shows amplification or translocation in glioma and leukemias, and receptor for KIT ligand shows point mutation and is seen in gastrointestinal stromal tumor, seminomas, and leukemias, while anaplastic lymphoma kinase or ALK receptor shows translocation in adenocarcinoma of the lung and certain lymphomas, while it shows point mutation in neuroblastomas. Similarly, aberration in the KRAS, such as point mutation, is seen in colon, lung, and pancreatic tumor. HRAS point mutation is seen in bladder and kidney tumors. NRAS point mutation is seen in melanomas and hematologic malignancies. GNAC point mutations are associated with uveal melanoma, and GNAS point mutations are seen with pituitary adenoma and other endocrine tumors. The non-receptor tyrosine kinase, proto-oncogene ALB, shows translocation in chronic myelogenous leukemia and acute lymphoblastic leukemia. The RAS signal transduction such as BRAF shows point mutation and is associated with melanomas, leukemias, colonic carcinoma and others. Notch signal transduction, Notch 1 shows point mutation and translocation and is associated with leukemia, lymphoma and breast carcinoma. JAK state signal transduction pathway, proto-oncogene is JAK2, which shows point mutation and translocation and is associated with myeloproliferative disorder and acute lymphoblastic leukemia. The nuclear regulatory receptor or nuclear regulatory proteins that are the transcriptional activator, MYC is translocated in Burkitt's lymphoma, while NMYC is amplified in neuroblastoma. The cell cycle regulator aberrations are also seen and associated with tumors. The series ND1 or cyclin D1 proto-oncogene is translocated in mantle cell lymphoma and multiple myeloma while it is amplified in breast and esophageal cancer. Cyclin dependent kinases CDK4 proto-oncogene is amplified or point mutation is seen in association with glioblastoma, melanoma and sarcomas. The oncoproteins accelerate the DNA replication and cell cycle progression, while tumor suppressor inhibit the cell cycle progression and DNA replication. This cell division is not merely DNA replication. It is basically the division of the cell into two daughter cells, and each of these daughter cells is a complete cell containing DNA and RNA, all the cellular organelle, cell membrane, etc. So there are many other oncoproteins or pathways are also involved in the division that coordinate the biosynthesis of all essential cellular component. So what is an oncoprotein and how it is involved in cell growth? A proto-oncogene may encode growth factor receptor, signal transducers, transcription factor or cell cycle component. When it is mutated, it becomes constitutively active and it is now termed as oncogene. Then the product of these oncogene is oncoprotein. The oncoprotein serve functions similar to their normal counterpart, but they differ from the proto-oncogenes that they have lost the off switch. They are constitutively active and thereby they are now independent of their dependency on the growth factor. There are many types of the receptors. Some may stimulate or inhibit proliferation. These are the cell membrane molecule that respond to ligand produced by other cell. The receptor for the steroid hormones are located within the cytosol. The receptor tyrosine kinase possesses intrinsic tyrosine kinase activity. They, after binding and recognition, they phosphorylate itself and other molecule. The non-tyrosine kinase receptor undergo structural rearrangement, making them receptive to initiate downstream signaling. These type of the receptor, the non-kinase receptors, often associate with non-receptor tyrosine kinase, which mediate further signaling. This table is showing the types of signal transducing receptors that are important in tumorigenesis along with the prototypical ligands. The receptor tyrosine kinase ligand are epidermal growth factor, insulin-like growth factor 1 and insulin, while G protein coupled receptors ligand are prostaglandins, RANTES and SDF1. The nuclear receptors are androgen, estrogen and other steroid hormone. Serine threonine kinase ligand are transforming growth factor beta. Kinase associated receptor ligand are growth hormone, T cell receptor and interleukin 2. Extracellular matrix receptors ligand are fibronectin, collagen and laminin. This illustration is showing growth factor mediated activation of transcription with resultant cell growth in cancer. Growth factor receptors RAS, phosphoinositol 3 kinase, MYC and D-cyclin are oncoproteins that are constitutively activated by mutation in various cancers. Here you can see that growth factor has bound to receptor for growth factor, and it is resulting in activation of the RAS by phosphorylation of the GDP and association of a bridging protein. This active RAS goes down and this active RAS is responsible for activation of subsequent pathway that result in growth of the cell. There are two breaks in the system which are shown in this illustration. One is the GTPase activating protein, that is GAP, that apply brakes to the RAS activation, and another brake is the P10 or phosphatase and tensin homologue that serves the same function for phosphoinositol 3 kinase. So now we will try to understand the effect of growth factor and activation of the signaling pathway from the membrane to the nucleus and identify at each step along the way some of the genes that are most commonly dysregulated and hence responsible for oncogenesis. Most of the growth factors are usually produced by one type of the cell and they affect on the other type of the cell, and hence they have got paracrine action, but the tumor cell has the characteristic to produce both the growth factor and growth factor receptor, such as the glioblastoma often express both the platelet-derived growth factor and platelet-derived growth factors receptor. Similarly, many sarcomas overexpress transforming growth factor alpha and also they overexpress its receptor, that is the epidermal growth factor receptor. The interaction between ligand and receptor will result in response, and their interaction and the resultant response are variable, and many activated receptor tyrosine kinase and non-kinase receptors, they serve as a platform for other proteins including the non-receptor tyrosine kinase. This non-receptor tyrosine kinase phosphorylate itself or it can phosphorylate other non-kinase receptor that will result in the formation of a cell membrane complex, and this cell membrane complex will activate and engage the signaling intermediates. The engagement and recruitment of the signaling intermediates will result in activation of downstream signaling pathway. The receptor protein role is important in oncogenesis, as these receptors get rid of their dependence on the ligand. They become constitutively active and independent of activity or transformation by the ligand. The ligand and receptor activation will result in the activation of the receptor or conformational changes within the receptor. If these receptors are receptor tyrosine kinase and non-receptor tyrosine kinase, that will result in phosphorylation of the tyrosine. So these phosphotyrosines are recognized by the SRC homology 2 or SH2 domain on the signaling intermediates, and they bind together. So due to the binding and recognition of the tyrosine phosphate or phosphorylated tyrosine with the SRC homology 2 or SH2, there is activation of the downstream pathway, and the response is variable dependent on the type of the receptor that is activated. This SRC homology or SH2 is specific for individual protein that is bearing the phosphotyrosines. The growth factor receptor is a transmembrane receptor, and when it binds to its ligand, it dimerizes, and then this dimer is phosphorylated by the conversion of ATP to ADP. When it is activated, it attracts the inactive relay protein, and this phosphotyrosine now bind with the SH2 domain of the intermediate these relay protein, and that will result in the cellular response. In carcinogenesis, these receptors become independent of the ligand, and they exhibit growth factor independent tyrosine kinase activity. This concept is elaborated in this video link that is also shared in the references of this video. There are various growth factor receptors that are epidermal growth factor receptor family, FMS like tyrosine kinase 3, receptor for neurotrophic factors, platelet derived growth factor receptor, receptor for KIT ligand, and anaplastic lymphoma kinase receptors. The epidermal growth factor receptor is by ERB gene, ERB proto-oncogene, the ERB1 that is erythroblastic oncogene B1 encodes epidermal growth factor receptor, and if this is mutated, it will result in adenocarcinoma of the lung. The erythroblastic oncogene B2 encodes HER, and if it is amplified, it will result in breast carcinoma, or it is seen in association with the breast carcinoma. The FLT3 point mutation or small duplications are seen in leukemias, while point mutation in the rate that encodes the receptor for neurotrophic factor will result in multiple neuroendocrine neoplasia 2A and B and familial medullary thyroid carcinoma. The platelet derived growth factor receptor B amplification and translocation will result in glioma and leukemias. The proto-oncogene KIT forms receptor for KIT ligand, and if it is point mutated, it is seen in gastrointestinal stromal tumor or GIST, seminomas and leukemias. Anaplastic lymphoma kinase receptor is encoded by the proton gene of this is anaplastic lymphoma kinase gene, and when it is translocated, it will result in adenocarcinoma of the lung and certain lymphomas, and if shows point mutation, it is associated with neuroblastoma. The anaplastic lymphoma kinase, when there is deletion on chromosome 5, it will result in fused part of the ALK gene with part of another gene, EML4 gene. So this fusion gene formation will result in the production of a chimeric protein. This chimeric EML4 ALK protein is there, and there is also constitutive activation of the tyrosine kinase. Identification of the receptor autonomy and products resulting in mutation has helped in therapeutic intervention, as antibodies and various drugs have been developed to block the activity of these mutated kinases. Such as HER2 blockers are helpful in breast carcinomas. Similarly, inhibitors of epidermal growth factor receptor and inhibitors of anaplastic lymphoma kinase are useful in patients having the mutation or fusion proteins in the adenocarcinomas, and these therapeutic targets, they block the tumor growth and also they induce apoptosis and result in tumor regression. However, they are not helpful in advanced cancer, as there is presence of acquired mutation in the advanced cancer. The important member of the RAS family are KRAS, NRAS and HRAS, and these are guanine nucleotide binding proteins, and they are activated by tyrosine kinases, commonly with the linker protein GRB2. They bind with GTP and GDP both. When they are bind to the GTP, guanosine triphosphate, they are considered as active in form, and this activation will result in downstream signaling and cellular proliferation, and it is limited by gap, that is GTPase activating protein, and in GTP form, guanosine diphosphate form, it is inactive. The binding with the GTP is catalyzed by GEF. When there is a mutation in the RAS, any member of the RAS family, it will block the activity by GTPase activating protein, and this will result in constitutively active downstream signaling of the by this GTP. So it will result in malignant tumor formation. So this mechanism is involved in activation of many G protein coupled receptors, and the proteins are different. Unlike RAS, these G proteins tend not to be mutated in cancer, but they may be overexpressed, and hence they achieve the desired effect, that is the constitutive activation of the downstream signaling. This illustration is showing the mechanism of action of the RAS. So the diagram A is showing the normal pathway, while the B is showing the pathway in the mutant cell. The step 1 is showing that RAS is inactive with GDP, and when it is facilitated by GAF2 and GTP, it turns into GTP RAS, and GTP RAS then activate the GTPase activating protein, and GTPase activating protein has got two functions. One, it transforms GTP into GTP, hence inactivate the RAS, and also it initiates the downstream signaling and activate the proliferation of the cell, so effector mechanism is started. While in the diagram B, it is clear that the off switch is blocked by mutation, so it will result in continuous stimulation of the cell that result in the exaggeration of the effector response, and subsequently the cell is transformed. Now the components of the receptor tyrosine kinase. You can see here that the RAS is activated, and downstream this RAS has got two arms. One is the RAF and another one is the phosphoinositol 3 kinase AKT and mTOR. So there are two arms of the RAS which are responsible for downstream effect. Usually there is gain of function mutation in the RAS, and this gain of the function, if it is there, the activating mechanism in the receptor tyrosine kinase almost always absent. The lung adenocarcinoma fall into mutually exclusive molecular subtypes that are associated with mutations involving RAS and various tyrosine kinase genes which implicate in targeted therapy. The most common type of the mutation is point mutation, and in this RAS, and as we have seen earlier, there are three members of this RAS family: HRAS, KRAS and NRAS, and these are mutated in 15 to 20% of all human tumors, while the RAS mutation is present in 90% of pancreatic adenocarcinoma, and half of the colon, endometrial and thyroid cancer show this RAS mutation, and 1/3 of the lung adenocarcinoma and myeloid leukemia, they exhibit this mutation. There is one important break in this RAS pathway or RAS signal transducing pathway, that is gap protein. This gap, that is the GTPase activating protein, it stops the activation of the RAS by converting the GTP into GTP and inactivate the RAS. But if up till now we have seen that there is gain of function of mutation in the RAS, but if this GAP, this gap protein is lost, this is the loss of function mutation. If this loss of function mutation, this gap protein that is inhibiting the activated RAS, so there will be persistent activation. So this inactivation of the GTPase activating protein will lead to the permanent activation. This gap protein is encoded by neurofibrin gene, NF1 gene and neurofibrin gene, and this loss of function will result in the persistent activation of the RAS signaling pathway, as happens in neurofibromatosis type 1. The phosphatidyl inositol 3 kinase are activated by receptor tyrosine kinase and G protein coupled receptors, and they phosphorylate phosphatidyl inositol lipids and are responsible for the formation of phosphatidyl inositol 3 phosphate, PI3P, or phosphoinositol 3,4,5 triphosphate. The PIP3 is a heavy molecule while PI3P is a small molecule, and these mediate many proliferation related and cell survival reactions. The phospholipase C is also activated by G protein coupled receptors and other receptors, and it divides the phosphatidyl inositol lipids into inositol phosphate signaling intermediates and diacylglycerol. Both of these they are responsible for the cellular multiplication. The inositol phosphate signaling intermediates through calcium signaling pathway and diacylglycerol through protein kinase C pathway are responsible for multiplication of the cell. The mitogen activated protein kinases or MAP kinases are activated by RAS following the receptor tyrosine kinase activation and G protein coupled receptors, and by other mechanisms. Some very important driver mutations such as BRAF occur among these proteins, and they frequently lead to the constitutive activation. Typically, these MAP kinases involve three sequential species, one activating the next, and the consequences of MAP kinase stimulation are diverse, and there is extensive cross-linking between these and other signaling intermediates. The downstream components of the receptor tyrosine kinase signaling pathway are also involved in the carcinogenesis or oncogenesis. The MAP kinase or mitogen activated protein kinase and phosphatidyl inositol 3 kinase and AKT cascade mutations are frequently encountered, and there is presence of a series of kinases with gain of function mutations, and these gain of function mutation will result in the phosphorylation and then the cellular proliferation. One of these is mutation in BRAF, that is, this BRAF stands for rapidly accelerating fibrosarcoma, and this BRAF gain of function mutation is seen in 100% of the hairy cell leukemia, 60% of the melanomas, and few other neoplasms including colonic carcinomas. The activating mutation in BRAF stimulate downstream kinases and activate transcription factor that results in the proliferation of the cell. The mutation in other MAP kinase family members downstream of BRAF are less common in cancer. This finding suggests that mutation affecting factors near the top of the cascade are most effective at producing the pro-growth effects. The mutation in the kinases of phosphatidyl inositol 3 kinase family are also seen, and the gain of function mutation is seen in 30% of the breast carcinoma. The break to this phosphoinositol 3 kinase is P10, that stands for phosphatase and tensine homologue. This P10 is negative regulator, and it's a tumor suppressor gene. So its loss of function of this P10 is seen in endometrial carcinoma. After receptor tyrosine kinase activation, phosphatidyl inositol 3 kinase is recruited to the plasma membrane, and with its associated protein complexes, there is activation of cascade of serine threonine kinases, including AKT. AKT is also termed as protein kinase B that phosphorylates more than 150 proteins. There are many substrates for the AKT. The substrates are key regulators of protein synthesis, mTOR and apoptosis, BAT, FOXO transcription factor, MDM2 and IAP. The RAS protein or RAS pathway is involved in the cancer so commonly that various attempts have been made to target some of the protein that are involved and are characteristic of this pathway. The GTPase activating protein is impossible to achieve its or regain its function as it is lost frequently, and some tumors such as melanoma are successfully treated by blockers of BRAF and this BRAF blockers are successful in the tumors or in the melanomas that exhibit the BRAF mutation, and this type of the treatment is not recommended for the tumors that are containing wild type of the mutation. Similarly, mutations in the phosphatidylinositol 3 kinase are also involved in the oncogenesis, so blockers of this protein are also being successful in the treatment of the cancer. The transforming growth factor beta or TGF beta is an extracellular cytokine that is present in the microenvironment of the cancer cell. It is example of cell communication mediator, and it triggers important regulatory pathways, and hence it influences the pathogenesis of the tumor. The response of this cytokine is contextual. In normal cell, it suppresses tumor development by modulating cell proliferation, survival, adhesion, and differentiation. Also, it inhibits mitogenesis that is induced by constituents of the extracellular matrix, while in tumor cell there is abnormal signaling in transforming growth factor beta, and it stimulates proliferation of tumor cell, facilitate evasion of host defense mechanism by the tumor cells, and promote invasion and metastasis. The tumor cells do so by mutation in the genes of transforming growth factor beta receptors and through interference with the downstream signaling by mutation or by promoter methylation of key proteins. These mechanisms hijack the regulatory activities of the transforming growth factor beta and adjust them according to their need, such as tumor growth, invasion, and metastasis. These mutations or interference are seen in cancers of colon, prostate, stomach, breast, pancreas, ovary, and many others. The granulocyte macrophage colony stimulating factor and interleukin 3 are other cytokines, and these produce their effect simply by overexpression, and they are commonly seen in hematopoietic malignancies. This table is summarizing the effect of the transforming growth factor beta normally and during oncogenesis. Normally, the transforming growth factor beta promotes apoptosis, promotes differentiation, and also promotes maintenance of the cell number, while it inhibits inflammation and mitogenesis induced by extracellular matrix. When there is failure of the tumor suppression, or when there is change or mutation in this transforming growth factor beta, there is promotion of the autocrine mitogenic activity and motility, while the immune surveillance is inhibited. They are responsible for the recruitment of myofibroblast, the extravasation of malignant cell,
modification of microenvironment and mobilization of the osteoclast during oncogenesis. The estrogen and progesterone are the steroid hormone that binds to specific receptors within the cytoplasm, and with the binding they form hormone receptor complex. These hormone receptor complexes are then transferred to the nucleus, where they initiate transcription that promote proliferation of the cells. So this is the mechanism of the action of most of the lipid soluble or steroid hormones. Other nuclear receptors are androgens, corticosteroid, vitamin A and D, fatty acid and dietary lipids. The high incidence of breast cancer in nuns was noticed by Ramazzini 300 years ago, and now it is clear that it is due to unopposed estrogenic stimulation of breast epithelium receptors and progesterone that bind to specific cytoplasm receptors. Anti-estrogenic therapy of the hormone receptor positive tumor cells reduces the risk of recurrence after surgery in breast tumors. The effect of androgen in the case of prostate cancer is very well studied, and the prostate cancer growth is stimulated by binding to androgen receptor, and this pathway involves or interact with many other cellular pathways such as cell cycle pathway, apoptosis pathway and differentiation pathway through various cytokines such as epidermal growth factor, insulin-like growth factor 1, fibroblast growth factor, vascular endothelial growth factor, transforming growth factor beta, and many others. So when this androgen is inhibited by surgery or drug, that will lead to decrease in growth of the prostate cancer. But eventually, through various mechanisms, the prostate cancer, however, it become hormone insensitive. Earlier, it was considered that the mucins are the glycoproteins that separate the extracellular and intracellular environment, but now it is well understood that these are the family of transmembrane glycoprotein that have got extracellular and cytoplasmic domains. The extracellular domain lubricate and protect the cell surface, while the cytoplasmic domain provides a scaffold for the interaction with signaling molecule that influence the cell proliferation and survival. The mucin 1 is increased in some malignancies such as breast cancer and malignancies of colon, ovary, pancreas and lung. This picture is describing the location and the structure of the mucin. The membrane bound mucins, they have got an extracellular domain and the cytoplasmic domain. The extracellular domain is providing the lubrication, and it is separating the both the environments, while the intracellular domain or the cytoplasmic tail is providing the scaffold to the attachment for various factors that are involved in various signaling pathways. Whatever the mechanism, either the receptor ligand interaction or constitutively activating driver mutation, there is constant interaction among the intermediate signaling pathway, and result of unrestrained activation of a given gene are not always predictable as a result of continuous crosstalk between these intermediate signaling pathways. So a mutant gene may produce different effect in different type of the cell. such as in this mutant gene in this cell A will result in proliferation, while in another cell it will result in apoptosis, and in the third type of the cell, cell C, it will result in differentiation. So the results of the mutant gene are not always predictable and are exclusive for that specific type of the cell. The non-receptor tyrosine kinase are located within the cytoplasm and within the nucleus, and when activated, they activate same signaling pathway as receptor tyrosine kinase. In most of the instances, there is chromosomal translocation or chromosomal rearrangements that result in formation or development of fusion gene. These fusion gene formation is responsible for constitutively active tyrosine kinase, and there is one example of this activity that is seen in chronic myeloid leukemia and subset of acute lymphoblastic leukemia, in which this ABL gene is from the chromosome number 9 is translocated to BCR locus in the chromosome 22, and this translocation and fusion with the BCR gene forms a fusion gene, that is ABL BCR fusion gene. This fusion gene encodes a fusion protein that is the ABL BCR fusion protein. This fusion protein is constitutively active tyrosine kinase, and the BCR part, the BCR moiety or BCR part is the non-tyrosine kinase partner that promotes its association with ABL, and it drives the ABL tyrosine kinase activity. So when this tyrosine kinase is active, there is activation of the growth factor signaling pathways. This tyrosine kinase activity can be inhibited by inhibitors of tyrosine kinase. Many different tyrosine kinase consist of a chimeric protein in which non-tyrosine kinase partner drives self-association with the tyrosine kinase. BCR-ABL chimeric protein is the characteristic of chronic myeloid leukemia, and this in this chronic myeloid leukemia, this chimeric protein is responsible for all the activation of all the signaling mechanism. This is an example of oncogene addiction that describes that tumor cells are highly dependent on the activity of one protein. So if BCR-ABL inhibitors are administered, that will block the activity of this chimeric protein, and the structure that was standing on this activity by the chimeric protein, that is the signaling mechanism, is collapsed completely. So it will result in the further tyrosine kinase activity and cellular proliferation and other activities. But interestingly, it is seen that the administration of this inhibitor does not lead to full cure from this chronic myeloid leukemia, but the proliferating component of the tumor is suppressed, and it is due to presence of chronic myeloid leukemia stem cell that harbor the fusion gene. So the patient should receive indefinite therapeutic administration of the BCR-ABL blocker. If it is discontinued, the stem cell, they will proliferate rapidly and they will give offspring, and the full-blown leukemia will recur. The other mechanism by which the non-receptor tyrosine kinase are mutated is point mutation. In these, the in these, the point mutation overturns the function of negative autoregulatory domains that normally hold enzyme activity in check. One example is the JAK state signaling pathway, in which the growth factors and non-receptor tyrosine kinase are responsible for the transduction of mitogenic signal. When this JAK state signaling pathway is activated, it alters the expression of the target genes that binds state transcription factor. One component of this signaling pathway is JAK2, and this JAK2 is also a non-receptor tyrosine kinase, and it has got activated mutation. If it gets an activated mutation, it will lead to its independence from the activation of the growth factor and receptor tyrosine kinase, other stimulatory factor. So now it is independent of the stimulatory factor. It is seen in myeloid neoplasm, in which it is now independent of the erythropoietin activity, and it function itself and is responsible for the transduction of the mitogenic signal. This finding led to the development of inhibitors of the JAK2 that are used therapeutically in several myeloid neoplasms. The ability of the cancer cell to proliferate is dependent on the group of the gene or gene that is turned on or off by the mutation, and if there is upstream mutation, it will result in production of the wild type of the protein, excessive production of the protein, and sometimes the transcription factors are also mutated, and the transcription factor mutation will result them and will give them under the control of the more vigorous promoter that result in overexpression of that transcription factors. The important transcription factors are MYC, FOS and JUN, and androgen and estrogen receptors. The MYC is a ubiquitous transcriptional factor, and it controls about 10 to 15% of all human genes. The MYC family contains C, N and L members, and there are many functions of this MYC transcription factor that have been described. They push cellular proliferation. Also, they favor stemness and they increase energy production. They also facilitate tumor cell invasiveness and activate cell death program in the cells that have intact p53 and other cell death effector mechanisms. This MYC is a key to development of many tumors. Sometimes the dysregulation involves genetic alteration of MYC itself. These can be translocations as is seen in Burkitt's lymphoma and subsets of other B and T cell tumors. Similarly, gene amplifications in the carcinoma, breast, colon, lung and other tissues, and gene amplification in the other members of this MYC family, the N-MYC, that is seen in neuroblastoma, while amplification of L-MYC is seen as the small cell carcinoma of the lung. In many instances, this activation of MYC protein and increased amount of the MYC protein is due to upstream mutation of the signaling pathways, and these result in increasing MYC transcription, enhancing MYC messenger RNA translation, and stabilizing MYC protein. There can be constitutive RAS, MAP kinase signaling, NOTCH signaling, Wnt signaling and hedgehog signaling. All these signaling pathways are involved, and finally they elevate or increase the concentration of MYC, hence they are responsible for the oncogenesis. The single nucleotide polymorphism is also associated with oncogenesis, and it is seen in prostate and ovarian carcinoma, and in certain leukemia the enhancer element are present with the MYC, and these variants, they appear to stimulate the activity of the MYC RNA expression in response to growth promoting signals. This is an example of the amplification of the N-MYC gene in the human neuroblastoma. In this picture it is seen that the N-MYC gene that is present on the short arm of the chromosome number 2, either it becomes amplified and it is known as homogeneous staining region or HSR region, or it is seen as extra chromosomal double minute, and the integration of the gene involve other autosomes such as 4, 9 and 13. It is another example of the MYC gene dysregulation. In this example, that is the Burkitt's lymphoma, this MYC gene is translocated from chromosome 8 to chromosome number 14, and it is placed near the immunoglobulin gene, and so there is increased MYC protein production and increased expression of the pro-growth gene. So this is the example of rapidly growing tumor, that is Burkitt lymphoma. The FOS and JUN transcription factor, they form the AP1 transcription factor complex, that is activating protein 1 complex. It binds with the DNA and is responsible for various functions, such as it promotes cellular proliferation and survival, and also it represses expression of such tumor proteins such as P53. As we have seen earlier, the estrogen and progesterone bind to the estrogen and progesterone receptors and make a hormone receptor complex, and this hormone receptor complex is then transferred into the nucleus where it is responsible for their transcription. But these ER and PR, the estrogen receptor and progesterone receptor, themselves act as transcription factor also, and they promote proliferation of the cell and depending on the cell type. For example, the mammary epithelial cell proliferated by estrogen receptor. Similarly, the prostate tumor proliferated by the activity of androgen receptors. These are independent of the hormone activation, and hormone is not necessary for this transcription activation. And there is another mechanism when these receptors are produced by the tumor cell itself, and it leads to autocrine activation. So as the hormone receptor complex is not making or not is formed in this mechanism of the transcriptional factor, and these ER, PR and the androgen receptors, they work themselves as the transcription factor, so there is no role of the hormone antagonist in the therapy of these cases. As we have seen earlier in the video of the cell cycle from normal towards the oncogenesis, the growth factor, they are responsible for the signal transduction and stimulation of orderly progression of the cell through various phases of the cell cycle. It is dependent on the binding of the cyclin dependent kinase with the cyclin. The binding with the cyclin activates the cyclin dependent kinases. These are described by Timothy Hunt in 1982, and these are named after the hobby of the scientist, that was cycling, and they are named after his hobby as cyclin.
and also they are cyclic in nature during their production and degradation, hence they may be termed as cyclin. So these kinases, the cyclin dependent kinases, are then responsible for the phosphorylation of the target protein that is responsible for the cell cycle progression. Once these cyclin dependent kinases are activated and they are responsible for the cell cycle progression, there is check on these cyclin dependent kinases, and that check is applied by the cyclin dependent kinase inhibitors, CDKIs, that blocks its activity. And when the mitogenic signaling pathways are activated, they block the activity of CDK inhibitors, so cell cycle progress continuously, or cell cycle progress as desired. This table is summarizing the cell cycle component and their main function that we have discussed already in detail in the first video on this molecular basis of cancer series. The cyclin and cyclin dependent kinases, the important component is CDK4 and D cyclins. They form a complex that phosphorylates RB, allowing the cell to progress through the G1 restriction point. The cell cycle inhibitor is CIP-KIP family, and P21 and P27 are important members. They block the cell cycle by binding to cyclin CDK complexes. P21 is induced by tumor suppressor P53, while P27 respond to growth suppressors such as transforming growth factor beta. The INK4 and ARF family, that is also termed as CDK and 2A to C, and they have two members. One P16 INK4A bind to cyclin D CDK4 complex and promotes the inhibitory effect of RB. P14 ARF increases P53 level by inhibiting the MDM2 activity. The cell cycle checkpoints components are RB and P53. Both are tumor suppressors, and the RB has got tumor suppressive pocket protein effect that binds E2F transcription factor in its hypophosphorylated states and prevent G1 to S transition. It interacts with transcription factor that regulate differentiation, while P53 is another tumor suppressor that altered in the majority of the cancer, and it is induced by DNA damage, causes cell cycle arrest by regulating the CDK inhibitor P21, and it induces apoptosis by upregulating BEX and other pro-apoptotic genes. So this is the summary of some important events that we have earlier discussed in detail, and this transition from G1 to S occurs when the activity of complexes involving cyclin A and E overwhelm the inhibition by CYP and KIP family of inhibitors, and the G2 to M transition occurs when cyclin B CDK1 complexes, cyclin B CDK1 complexes are activated by removing inhibitory phosphorylation of the CDK1. So this 1 and 3 are activated by this checkpoint 1 and 3 are activated by DNA damage, and 2 and 4 are activated by other factor. This is the background knowledge. For the pathology we will just see. The cancer associated mutations that affect the G1 by S checkpoint, they
can be divided into two groups. One is the gain of function mutation in D-cyclin gene and CDK4, which promote unregulated G1 by S progression, and therefore they function as oncogenes. There are three types of the D-cyclin gene, D1, 2, 3, and they function interchangeably, and they are often dysregulated by acquired mutations in cancer. There may be presence of chromosomal translocations such as that is seen in the lymphoid tumors. Gene amplification is also seen in some solid tumors. CDK4 gene amplification is associated with melanomas, sarcomas, and glioblastomas, and CDK4 inhibitors are effective in treatment of advanced breast cancer associated with excessive CDK4 activity. The mutation that affect other CDKs and cyclin E are infrequent, and they are less important in the control of G1 by S transition. The other group of the tumors associated mutations that affect G1 by S checkpoints is the loss of function mutation in genes that inhibit G1 by S progression. These are CDK inhibitors, which inhibit cyclin D CDK complexes and are therefore mutated and otherwise silenced in many human malignancies. Germline mutation of P16 are present in 25% of the melanoma prone types, and somatically acquired deletion or inactivation of P16 is present in different percentage in various tumors, such as it is present P16 deletion or inactivation is present in 75% of the pancreatic carcinomas and 40 to 70% of the glioblastomas, and half of the tumors from the esophagus they show this P15 deletion or inactivation. 20 to 70% of acute lymphoblastic leukemia also show this P16 inactivation, and P16 is also associated with non-small cell lung carcinoma, soft tissue sarcomas, and bladder cancer, in which about 20% cases show this deletion or inactivation. The two most important tumor suppressor genes, the RB and T53, they both encode proteins that inhibit G1 to S progression. This table is summarizing common proteins that derive cell proliferation, drive their activity and activation.